Hello everyone.
Hello.
Okay, we see some more people are coming in. If you could type in the chat where you're from, that would be great. Okay, maybe I can start. So I'm currently in the UK. I'm a PhD student at University of Oxford. Aarushi, what about you?
Yeah, I'm joining from Toronto. I'm a master's student at the University of Toronto. And yeah, it's pretty early in the morning here.
It's 4 a.m. Aarushi is very passionate about the brain, and so she woke up at 4 a.m., which is amazing. Hi, Tano. Hello. Hope
Hello.
you're doing well. So I think it's 9 a.m. here. Oh, we have a few more people. Okay, hello everyone. Welcome. It's great to see you all today in this session, and thank you again for coming. I'm Sapo. I'm a PhD student at the University of Oxford, and I'm also one of the co-chairs of the 2025 Fly Connectome Research Competition. And today I'm joined by Aarushi, who's also one of the FlyComp co-chairs and research lead. And today she would be the one carrying the conversations and the workshop today. We have this exciting data, the CNS Connectome of the fly, and as you will hear from Aarushi, it's an exciting data set, and it's also relevant to the competition and the tools that you might be using for the competition. Before we start, I want to thank the audience for being here today, and I'm so excited to present some of the groundbreaking work that folks have been doing in the field of synapse-level connectomics. And if the audience, if you would like to put in the chat where you're from, so that would be great. So Archie, would you like to introduce yourself and then maybe get started?
Okay, sure. So hi everyone. It's very nice to see everyone here joining us today. I'm particularly really excited to give this workshop with Sapo because this is a very, very exciting data set. So far we've been talking about the optic lobe. We also have a central brain data set, but as you can see from this beautiful a video over here, we now have the entire central nervous system of the fly. So you can think of this as a brain and body. So we should totally dive into it now. So let's get going. So what is a connectome? So Sapo, you can lead this part.
Yes. So you might be like, what is connectome? And I want to say that connectome or connectomics actually is the production and study of connectomes. And the connectome itself is basically brain map. But why would neuroscientists spend so much time in understanding brain map? And the answer is that you might be like, that's it, brain map? And there's this quote that I wanted to share, and I think it really highlights the importance of understanding the brain map or how the neurons connect to each other. So there's this quote by Dr. Joshua Wilkinson from Johns Hopkins University. So he says, if we want to understand who we are and how we think, part of that is understanding the mechanism of thought, and the key to that is knowing how neurons connect with each other. And essentially, what he's saying is that if you understand the wiring diagram of the brain, we understand the mechanism of who we are as a person and how we think. And how we think means this could mean like how do we memorize things? Why do we like eating chocolate compared to eating something bitter? Or why do we get scared of snakes? So how we think is really important. And also what happens when things go wrong in how we think, such as in neuropsychiatric disorders or neurodegenerative disorders. And that's why the study of connectomics is fundamental to neuroscience and understanding of our brain and cognition. So what exactly are brain maps? When you think of brain maps, there are two main things to consider. The first is called a scale. So there are two kinds of scale. One is microscale and the other is macroscale. So when you think of MRI scans, that's macroscale. It's macroscale because you're imaging the entire brain, which is fine, but it's macroscale because at least in the humans, when we take MRI scan, you're not able to see at the level of synaptic connections. So in other words, when you take the scan of our brain, you're not able to tell or you don't have the information of all the billions of neurons that exist in the brain. Then there's the second kind of scale, which is the micro scale. So in the micro scale, or we call this synapse level connectomes or synapse level brain maps, you can actually see the brain map at the detail of the individual levels of neurons. So in other words, you can actually count how many synapses does neuron A connect to neuron B. And for our competition, we are using the Fly Synapse-Level Connectome, which Aarushi will go over. And here you can actually explore the individual neuronal connections and so on. The second point to consider when you think of brain map is whether this is a structural map or a functional map. So a structural map is, of course, you're studying the neuroanatomy. So the field of synapse-level connectomics is really huge in trying to understand, let's say if you have neuron A and neuron B, how many synapses does neuron A connect to neuron B, or how many points of connections there is. In addition to structure, you can also try to infer a function. So at both micro scale and macro scale, you can sort of measure the brain activity, of the functional dynamic of individual neurons, of the individual brain regions. So in the fly, you have calcium imaging. So you kind of try to image the functional property of certain regions or certain neurons. And then in humans, you might have heard of functional MRI, where you infer the function from the blood flow, because you assume that when a part of the brain area is active, you get some blood flow to energize that certain region. And because there's an inherent change in the magnetic property of the blood, you can infer some sort of function. But for the purpose of today's presentation, we'll be focusing on the synapse level connectome of the fly, which is a groundbreaking dataset and a very recent breakthrough in neuroscience, because now, for the first time, you can sort of see the level of detail in the fly brain. And today you're going to see from Aarushi how exciting that is. So yes, I will let Aarushi take on the Fly Connectomics. As you can see, there's such beautiful imagery. So Aarushi, please take it on from here.
Yeah, so I'll be talking now all about Fly Connectomics. And if you've been watching our previous workshops or you're already working on the competition, this might be a bit familiar with you, but if you're new, we'll carry you from the start. So, what is a connectome? So, as Sapo's already gone over, but just again a refresher, I guess, it's a structural map of neuronal connections in the brain, but synapse level resolution connectomes have only so far existed for 3 species up till now, with the most well-known being that of a tiny little worm that we call C. elegans, and this has around 300 neurons. Last year, we got a full-brain adult Drosophila melanogaster or fruit fly connectome, as you can see from this video here. And it is the largest synapse-level connectome of a full brain of an entire organism. And this data set has 50 million chemical synapses with 140,000 neurons. So what is a synapse-level connectome? So to give you a bit of intuition behind it and sort of an appreciation, essentially, it is how two neurons connect at the synapse level. So the gap between the two neurons would be the synapse, right? And again, as to give a bit of an appreciation for the complexity and the detailedness of these data sets, let's have a look at this type of neuron in the fly visual system, which is called LC11. The connectome can tell me who's talking to LC11, so who are the neuron partners that connect to LC11, or who LC11 is talking to, or who LC11 partners up with. And it also tells me by the proportion that these connections occur, as you can tell from these sundial areas that are allocated for these, how strong these connections are. Another level of detail that the connectome can give you, let's have a look at this middle panel here. So let's focus our attention on a type of neuron, which is called MI1. So from here, the connectome can tell me this neuron called, which we call MI1 in the visual system, is comprised of 1,000 cells or 1,000 neurons. So that's how much that makes up this one neuron called MI1. Also, in specific, I can tell the number of input connections that MI1 is getting is like somewhere a bit less than 750,000. And the output connections it's sending is around 2.15 million. So that's another level of detail we can extract. And also, if you guys have been playing with NeuroGlancer, this looks familiar. You can actually look at these neurons, and you can also visualize where those synapse sites are also the predicted neurotransmitter identity, which I guess you can think of are the chemical messengers with what neurons communicate with. So you might be asking, okay, this looks really cool, but what's the point of all this? And do connectomes even help us in the first place? So I would like to motivate this talk by sort of linking to a big picture in neuroscience. One of the main goals in neuroscience is to understand how structure connects to function. It's really that. How does the structure of the neuron lead to its function? And if you really think of it, that's pretty cool that neurons do this. So you already know one part of this equation. And one part of this equation is the synapse level connectome. It tells us who connects to who at a very detailed resolution. And in Drosophila, we have this really cool, powerful genetic toolkit, which can manipulate neurons, like single neurons, and reveal its activity or like how it behaves, I guess you could think of. So I have this information of who connects to who's talking to who, and I also have this information of what a neuron is doing at a point in time. I can extract that functional information, you could say. And we could link these together, and somewhere in the future, this is the goal, we can extract how the fly produces really complex behaviors, which is actually already being undertaken. But I guess one of those goals is to now use these maps, these detailed, tools to manipulate neurons to get a very strong and detailed understanding of how the brain is so cool and how it does what it does. So I've been talking all about flies, but there's some really exciting progress in the connectomics field. This year we got a 1 millimeter cube, just to be exact, that's the grain of sand, a connectome of the mouse visual cortex. So as you can imagine, a mouse, you don't have to have work with them, but their brain is a lot bigger than 1 millimeter cube. So going up to synapse-level connectomes for much bigger animals is still underway and is a very exciting sort of thing to work on right now. And similarly, we also have the 1 millimeter cube of human temporal cortex, as you can see here. So yeah, so synapse-level connectomics is sort of now pushing for bigger species and with hopefully the goal of one day having a full connectome of the human. Now, I'll let Sapo take away this part.
Yes, so now that you know how fascinating this field of synapse-level connectomics is, and then it's a recent field partly because of the unprecedented AI tools that allow us to, image the individual neurons, which actually Aarushi will go over it very soon as to how technically challenging all of this is. I want to explain a little bit the motivation behind this competition, the Fly Connectome competition. When the groundbreaking data set of the Synapse-Level Connectome of the fly was released, Arishi and I were like, oh my God, these brain images look so beautiful. And you can actually visualize that using the tools that you'll be using for the competition. And not only it is beautiful, but it's something that's new, that's groundbreaking, and that's something we wish high school students and undergraduate students and young students get a chance to explore. And let's say you're interested in research, this is something that you can actually explore. And for instance, Arishi's thesis is literally exploring this data set that you'll be uncovering, and also our previous speakers who came and gave a talk. They also use these data set to explore the brain and also try to understand things like that. But essentially, to summarize it, very recently, in 2024 and 2025, neuroscientists from around the world have created essentially one of the most comprehensive wiring diagrams of the brain. And in other words, just a reminder, this is the study of how neurons connect to each other, and we call this connectomics. And we will be using this very fascinating data set to uncover new insights about the brain. And the data set that you'll be exploring is called the optical lobe, which has over 49 million synapses and 53,000 neurons. And can you imagine how massive this is? And together, Aarushi and I, on behalf of our faculty partner, Professor A.R. Gruntman, Ina Anreiter, and Steve Jordans, we would like you all to be a part of our learning journey to use powerful connectomics tools to visualize and analyzing connectomic dataset. And by studying neuronal connectivity, you can learn so much about the brain. And as you can see, we have two tools here. And all of this, by the way, is available in our website. But essentially, as a part of this competition, you'll get to learn how to use these two tools. And we have a very comprehensive guide and also previous workshop that kind of covers all of this already. So the first tool is called NeuroGlancer. So NeuroGlancer is a tool that allows neuroscientists and biologists to visualize individual neurons. And then you have another important, powerful tool called Neuroprint Explorer. This allows you to query neuronal connectivity. And also today, Aarushi might be using some of these tools like NeuroGlance especially to show you how the neurons look and so on. So this is very exciting. And the challenge is that once you use these connectomic tools to explore a neuron of interest in the brain, and Aarushi again will be showing what does it mean to explore a neuron. And then once you choose a neuron, you'll be exploring the structure or the neuron morphology. And then once you explore the morphology through visualizing these neurons, you can use Newprint to explore this neuronal connectivity. In other words, how does one neuron connect to the other? And finally, you will try to link structure to function, which is again the fundamental thing in neuroscience. So you're going to try and propose how your neuron of interest processes information and contributes to a behavior. And by engaging with these connectomic tools, you'll be able to do that. In the end, this comes down to two deliverables. So first is the creative piece, and the second is written research report. But again, all of the information is available online. So without further ado, Aarushi will explain this connectome. Actually, Aarushi, before you go over this, you should, I think, explain the optic lobe dataset and the entire brain. Maybe give that a bit of context if you are planning to go over it anyways, just so that people who might be new to our workshop will be able to understand how our competition kind of links to this dataset as well. That would be
Yeah, of
great. Thank
course.
you.
Of course. Yeah, so at the time, Sapo and I were launching this competition. This was only last year, by the way. We only had a very comprehensive detailed wiring diagram or connectome of the male optic lobe. And that's personally something I use a lot in my research. And that's what motivated our competition. And also, the visual system is just very beautiful in architecture, as in like a type of neuron would have many repetitions within itself. So this is something we call stereotypy. So like we can find like if you find something really interesting in one type of neuron and we see it in 100 of those type of neurons, it shows us that this is probably a legitimate biological thing that's going on. And that's something really cool about the fly's visual system. Only until I don't know if it was like 2 or 3 weeks ago that this preprint came out, which is the entire brain and body connectome of a male fruit fly. So we've had one of the female fruit fly, but this one actually has an intact neck connection from the body and the brain. And this is really exciting because as you can imagine, like think of yourself, you don't work in isolation, right? You don't just use your eyes, your brain's processing the information that you're seeing from your eyes. Similarly, even in my research, when I would just use the optic lobe, there would be a lot of really cool neurons that we would find that do some cool thing, but they would sort of truncate, or like they would sort of have weird like they wouldn't have the names of the partners they connect to in the brain, or they wouldn't have the right number of synapses because the data set just ended there. So this is really, really fascinating advance because now everything's connected to everything. So if you have a neuron of interest, you can see from the brain, from the optic lobe, from the sensory neurons, from the ventral nerve cord, exactly you can map out how that information is going. So even though you'll be using the optic lobe data set, that optic loop is from this fly brain. It's just back then they only completed reconstructing that part, and now we have the full thing. So if you're really interested in the brain, you can actually dive into this, and I'll be showing you how you sort of do that and explore things within this brain. So yeah, I hope that's exciting for you guys. So yeah, let's dive into the central nervous system connectome of the fly. All right, so as I mentioned, we now have a complete male Drosophila central nervous system, synapse-level connectome. In short, we call this male CNS, or the authors call it that. The data set has both the left and right optic lobes and the central brain, which is this part of the fly. So, and this also has the entire ventral nerve cord, which you could think of as the body of the fly. This data set has 166,691 neurons and 11,691 types. It is all proofread, all annotated. So massive effort and really cool resource. Also, as I mentioned, we previously had the female brain connectome. And now that we have the male connectome, we can use both resources to see whether there's similarities and differences between the male and female brains. This is something we call sexual dimorphism. So I've mentioned a lot of words. Let's dive in one by one what this means. So I said this connectome has been fully proofread. What does this mean? And I think this is something really cool about connectomics. So first things first, how do you actually get a connectome? What are the ingredients to get there? So one of that is you use something called an electron microscope. You literally shoot electrons down your sample. And you have a brain and you slice it like one by one, and you shine electrons and you image each of those layers. So this is an EM volume of a fly brain. And if you zoom in, the level of detail this data set gives you, as I mentioned, is like at the pre-synapse and all these post-synapses. So that's the level of data set you're getting. And you can imagine, wow, this is the fly brain volume, and this is the level of detail I'm getting. That is a lot of information, and you would absolutely be right. So remember, we have this EM data. I've cut it all up. I've imaged so many parts of this. But my goal is to actually get neurons, right? Like I want to see the neurons. Neurons are not like on a 2D slice. They're actually like 3D, and they would transverse through each of the slices I took. So one of those goals was, how do I get back from these 2D images to actual neurons of the brain? This is something we call 3D reconstruction of neurons. So there are advanced algorithms that are developed at Google, and with one being known as flood filling networks, to detect and segment neurons. Segment means reconstruct neurons in 3D. And these are from raw data. So essentially, this algorithm sees this raw EM data, and it reconstructs these 3D neurons. These are things we're seeing in neuroglands. But just like with a lot of programs out there, although fluffing networks are really great, they can make mistakes. And that's why we need to actually check and see whether what they segmented is correct. Remember, fly neurons are really complex. Here's an example of a CT1 neuron. Look, that's a lot of arborization. And these algorithms are amazing at detecting these, but that's why they can make some mistakes. And we need to check, did it get it right? So expert anatomists actually go in who are trained to check whether, things have been properly attached to the proper neuron, whether things are merged incorrectly or whatever. You can actually find YouTube videos on this process. It's very interesting. And essentially, this is really time consuming, and it's one of the more time-wise challenging things to do in connectomics. Of this particular connectome, the one of the male CNS, this was semi-automated and somewhat manual, but even then it was a 44-person-year effort. This is a way people describe how long it takes to, make a connectome. So basically it means it would take one person working full-time 44 years to actually proofread and get this out. So it was a lot of work. So yeah, essentially also one thing to know, the algorithm, as you can see, if you go on Neuroglance, it colors these up. So these are not actually the way you image them. This is the algorithm coloring different neurons. So we can differentiate now. Okay, so orange is a different neuron from the red one, essentially. So yeah, so that's what 3D reconstruction neurons are. And that's what proofreading is. Now let's go into cell types and annotation. This was another word I used. This connectome is fully annotated and there are cell types assigned. So annotations help the broader biology community, people like me, people like Sapo, and people like you who are research enthusiasts, better explore the connectome and have something tangible to relate to. So one way to understand this is when you get a connectome, you segment all those neurons, and it's great, you can see them, but there were in this data set 166,000 of those, and all of them are going to be assigned an ID like 1, 2, 3, 4, 5, 5, 6, 7, 8, 9, 10, 11, 12, 13. And as you can realize now that's very challenging to actually maneuver with, especially since neurons look pretty similar, they're very complex. So experts have actually gone in and actually made sense of this data. Like they've grouped things based on similarities, certain differences, and they've grouped them based on a broad overview characteristic into very, very detailed characteristics of neurons. So the authors of this paper have organized neurons in this connectome based on 4 classes. The first one is called superclass. Superclass tells us the direction of information flow, so for a neuron, and it depends on the location of the neuron and also its broad function. So for instance, this neuron in green here, this is a descending neuron. It goes from the brain to the ventral nerve cord. And in this class, there are 1,319 of those. So yeah, so that's a very coarse, very broad understanding of what a bunch of neurons in this class do. And they might be a slightly different level, but they overall share this descending characteristic. Then you have another one that's called hemilineage. This tells us something about the neuron's developmental origin. So this would be the hemilineage. So there are 54 neurons in this hemilineage, and they have a similar developmental or the same developmental origin. Before I jump into supertype, I'll go all the way to cell type first. So cell type, you might be somewhat familiar with, as we've covered in previous workshops, but essentially cell types is the most precise, fine-tuned way to group neurons. You group neurons based on how they look, which is morphology, and how they connect to other neurons. And most cell types in the fly visual system, the fly brain in general, have many neurons within it. So like, let's say my neuron I'm interested in is LC10A. LC10A will have it has 140 neurons that make up that cell type. And all 140 of those neurons connect exactly the same way, look exactly the same way. So that's cell type. But you see, there's a pretty big jump between hemilineage and type. So the authors put another classification, which they call supertype. So think about it. Let's say there's a neuron. There are a bunch of neurons that have the same hemilineage, but they're not just that. They also show pretty similar morphological patterns. So that's one step more detailed than hemilineage. But they differ in connectivity and precise morphology, so they're not really at cell type yet. So that's why they were made super type. And if you're wondering, well, why was that done? Because imagine if you went straight to the cell type level, these two neurons would be completely different, and you wouldn't be able to tell if they're related. They're just different. But the super type class helps us, connect that hemilineage with the type a bit more. So that's why that class exists. So a bit into hemilineage. So what is this? So the fly brain is built from a set of 120 neural stem cells, and that we call them neuroblasts. Each neuroblast you could think of has its own transcription factor toolkit. So it determines what kind of neurons it's going to produce and in what order they're produced. This neuroblast divides into two hemilineage, and both these hemilineages will have different morphology and neurotransmitter identity from each other. But the group of neurons in a hemilineage are going to have similar morphology, but the same neurotransmitter. One thing is, within a hemilineage, neurons grow together in a stereotyped hemilineage tract. Essentially, they pack into bundles and they cross the brain in very similar ways. And essentially, hemilineages are another way and a natural unit of functional and developmental organization of the brain. So that's another layer of information you're adding and another perspective you're looking at things from a developmental sort of origin. Now, going into cell type, and to help you appreciate why cell type is so powerful, this is something I use very heavily in my own research. And the authors of the study defined 11,691 cell types. Let's have a look at LC10A. So LC10 is an umbrella group of a cell type, and it has some number of neurons. Imagine now we didn't know that, okay? And I looked at these two neurons. They look pretty similar to me, and most likely to you guys, they look pretty similar to you. It's kind of hard to tell the difference. But the great thing about cell typing is that their experts have gone in, they found some difference in connectivity between these two, some difference in morphology, which gave it a good enough reason to group them into separate groups, LC10A and LC10B. So that is what cell typing is. Another terminology alert. I said sexual dimorphism, so what does that mean? And this is the main sort of advance that this paper has given us. So think about it. There are a lot of sex differences in behaviors between male and females and, Drosophila, mice, humans. And one part of that might be explained by sex-specific circuit diagram or like connectome of each of these sexes. And that's exactly what they did. They compared the female and male connectome datasets. So we had the female connectome of a brain from Flywire, and we have a hemibrain, partial brain of a female from here. And now we have the entire male CNS. So it's the perfect candidates to compare to each other. How do you find sexual dimorphism? So they compared using cell types. Cell types have really fine-tuned features, and they're fundamental unit of conservation across brains and hemispheres. So one hemisphere of the brain and the other. Also, as I've kept reiterating, most neuronal cell types contain many cells, right? So if I find a neuron that's different from the male, and this cell type, A, has 100 neurons in it, and 100 of these neurons in the female look different from 100 of the neurons in the male, then I'm more confident this is actually different between the sexes. Whereas I find just one neuron, different in the male and the female, then it's more likely there's just some artifact maybe in the data set, or maybe it's just a random connection. So that's why cell types are really helpful to find sexual dimorphism. And essentially, this analysis reveals shared and sex-specific features. So the authors grouped their findings into three groups through the comparison. One is isomorphic, as you can see here. These are a group of neurons with the same developmental origin, same overall, but not same connectivity and morphology. So, and an important thing is the number of neurons in each hemisphere have to be the same. Then they also have the sexual dimorphic category, which is the group of neurons in this category also have a shared developmental origin, but they exhibit some differences in morphology and hence, as a consequence, connectivity as well. And also the neurons have to be the same in both hemispheres. And then you have the sex-specific category, where a group of neurons in one sex, maybe the female, are not found in the male, so they're unmatched. And this is from both hemispheres. They have to be in both hemispheres for one sex, and they have to be absent in the other. And this one is only given if the neuron from the other sex, let's say the male, the most similar neuron to this category is already assigned a label. It's already assigned isomorphic or sexually dimorphic. Then we give it the sex-specific name because these variables describe that better than this one. So this is only given if it's missing in either of the two sexes. So an example of what sexual dimorphism and intuition behind this is, have a look at AOTU008. As you can see, this is a very beautiful example of how structure looks very different, right? So this is the male AOTU008. It's the exact same neuron, but you can see it has like, you call it feet. Looks like a bit of like a dog or something. But like it has like extra branches that are going down. As you can see, the female, she doesn't have, these neurons don't have those extra branches. Or if you can look, maybe as a more general example of how this looks, this is a neuron in one sex. A dimorphism, but maybe a sexually dimorphous neuron in the other sex might mean that neuron has an extra branch. Maybe it has more neurites, they're more dense, like if you compare them. Or, the axon dendrites are a little shifted from the original one, or it's just really complex from the other one. So this is actually how they're looking at sexual dimorphism, because if it looks different, it probably means they connect a bit differently. Overall, they found that the male visual system, which we're working with in our connectome, within our, competition, only 0.1 neurons are sex specific. So 0.1 of the neurons are present in the male and not in the female. Only 0.3 are sexually dimorphic. So there's very low. There's not much sexual dimorphism in the visual system. In the central brain, however, 3.4% of neurons are sex specific and 1.4% are sexually dimorphic. So they're different between male and females. And in the female specific neurons, only 1% are in the central brain. So only 1% of the central brain of the female are female specific. So now let's revisit this. So I said they're fully proofread. This connectome is fully annotated, and they describe 11,000-ish types. So that probably makes sense now. And we also compared the male and female connectomes, and we found sexual dimorphism. So that probably now will make sense. Now that we've talked about sexual dimorphism, let's have a explore how this actually looks. So I found the authors found this really interesting observation. It's pretty cool. So I think we should dig into this. And basically, they found, as I mentioned, that AOTU008 is a dimorphic neuron because it has extra branches in the male, but it doesn't have extra branches in the female, as you can see here. So they wanted to look at how this, as a consequence, what happens to the connectivity. In the male, they found that this neuron connects to a neuron called DNP05. This is an isomorphic neuron. Remember, alert, what does isomorphic mean? Isomorphic essentially means that overall, this neuron between the two sexes is similar in connectivity and similar in morphology. So it's the same in both data sets. And they looked at its connection to VEST02M, male-specific neuron. Alert, what does this mean? It means this neuron is only present in the male and it's not there in the female. And look how structure links to maybe function in this. And it's telling us a clue of the organization of the visual system. So see, in the male, when AO2U008 connects to DNP05, it's using those extra branches. As you can see, the synapses in yellow all localized it. It's using those extra branches to connect to DNP05. But then look, so it means, DNP05, remember, is present in the male and female, but in the male, it's connecting in a very specific way. There's a very specific way it's passing on information. So maybe in the female is different because the female doesn't have these. But in A02U008, which when it connects to VES202M, which is a male-specific neuron, this is only in males, it's not in females, you can see it's using its entire axon to connect. As you can see, the synapses are localized everywhere. So this is pretty interesting feature. So it means when it's connecting to a neuron which is only present in the male, this neuron sort of is not as specific. It's like, well, you're only in this, so I guess you can think of it, it sends everything it has to that. Because it's not in the female, but because this is in the female and the male, maybe that's where the sexual dimorphism in the behavior comes, whatever AO2U008 is involved in. Because in the male, it's connecting in this certain way, but in the female, it's a different way. So that's a way to link structure to function, I guess. And we can maybe test this out later experimentally. So now let's explore A02U008. Here's a QR link to the male CNS page. If you scan this, we can both explore together.
I put on a chat as well.
Okay, fantastic. So now we're gonna have an explore of this. You should have landed something that looks like this, male CNS connectome. If you scroll down, go to this page, male CNS connectome site, and open it in a new tab, and then go to Dimorphism Explorer that's down here. Since one of the biggest advances this work has also given us is that it's actually compared the male and female connectomes. So, they've told us about sexually dimorphic cell types or cell types that are male specific or female specific. Also, is not the only way to look at neurons. I mean, it's the only way to look at things at the synapse level, but people use light microscopy to actually visualize neuron much before this. And essentially, when they found these neurons, they had different names. As you can see, the author of the study and is given as well. So these are different names of neurons, and they've sort of also given a category of that. So if you read a neuron and you think, oh, that's so cool, I wonder what it's called in the CNS connectome. So they've all provided that information here. Also, you can sort by brain regions. If you like a particular, I don't know, internal lobe, for instance, you can find the neurons there. They're also grouped by supertype. This is, remember, the most coarsest, the most big level grouping of neurons where they share morphological features. They're not completely the same, but they're somewhat similar. And that you can see here. And they've also grouped cell types by hemilineage, as you can see here. So you can also have a look at that. So there are many different ways of, I guess, making sense of this data. So now my goal was to have a look at A02U008 is a sexually dimorphic neuron. So we'll go to the sexually dimorphic cell types and we'll scroll and we'll find it here. So you can see it tells us the superclass. So like, how is the information flow? So it's intrinsic means most of the information in the neuron is intrinsic to the central brain. Tells you the hemby lineage, the supertype, and also synonyms, maybe that literature, that because people found this before through light data, that's what it's called, neurotransmitter identity, and the number of these in the hemispheres between male and female, and also who it connects to down here. Now, for our purposes today, we just want to visualize stuff. So let's open this on a new tab. And I recommend opening 2 tabs of this for a different purpose, because we're going to compare stuff, and it's a bit easier if your browser lets you do that. Okay. So here are these A02U008 sexually dimorphic neurons, and in the male and female. So CNS seg tells us this is the male neuron. This is from the male dataset. Flywire mesh tells us this is from the female. So pink is female, blue is male. That's an easy way to think. I'd like you to go up here in this tab here. It says show layers type panel and click on it. Maybe this looks familiar because you guys are all experts in Neuroglancer. You'll actually find how to search particular neurons. So for this one, I just want to see my CNS neuron. So if you don't want to see a particular mesh or CNS seg, you just click on it and it won't show it. Just click on it, it won't show it. So as you can see in the female, this is what the neuron looks like. In the male, you see there are extra branches here, which is consistent with what I was telling you. In the male, there are extra branches, which makes this sexually dimorphic. Now let's see how this connectivity plays out morphologically. Let's get rid of flywire meshes. We'll go here, and I want to search DNP05. So be very wary of capital letters here. So there are 2, one per hemisphere. As you can see, this neuron is connecting to those extra branches in the male. And if I turn it around, you can see that it's contacting these. Let's have a look at the female one. So let's get rid of this. Let's go here in this panel at the side. And if I want to access the female data set, if you're using a mouse like you do this click here, or you can do a double click on your laptop, whatever. So it'll make it all pink. So that's what tells us we're in the Flywire dataset. And again, we're going to search up the DNP05 and 2 of those. And let's see. If I look at this way, it looks like it's connecting to this. But if I rotate it, you see they're actually not connecting like that. It's kind of hard to see how they're exactly connecting. But as you can see from here, they are connecting through these extra sort of legs that it has. But this one does not have that. So that's very clear, this sexual dimorphism. And this probably influences the female's behavior and the male's behavior just purely because of this connection, which is really cool. Now, we can also have a look at the other part of this, just exploring. We can look at VES202N. So this neuron you won't find in Flywire, the female, because it's only in the male. But we can have a look at it here. So let's try VES202M. So there are many of those. And if we visualize them, it's kind of hard to see, but like maybe if we enable maybe the post, actually the post and pre is going to show everything. So that's not really helpful. But yeah, so it sort of connects to everything, but these sort of things are a bit harder to see because it's sort of all over the place, sort of. So yeah, that's something you can do if you're interested. You can actually like, see maybe in the optic lobe, your favorite neuron was cut off short. So that was a bit sad, but now maybe you can search that neuron in this data set and see what it looks like, figure out maybe are there any cool things these guys found about the paper, about the neuron. So yeah, that's really it from me. Yeah, and thank you so much for listening. If you have any questions, I am happy to take them.
Great, thank you so much. I also have one more link, which is if you could fill our survey, that would be great because that's a way for us to know if we are doing well in our workshop. And remember, this is all anonymous, so we will never know who responded. So please take a time to fill the anonymous survey, which is the last link I've sent you. I'm also checking the link. They look fine to me. And please let us know if you have any questions. My first question is, Aarushi, maybe if you can go to the actual optic lobe connectome, which is what we're going to use for the competition, and maybe show neuroglancer and Newprint as well.
Sure. I don't know if you've sent the link to the optic lobe, but even if you're on this page, you literally click there and you'll get
You
the
can
optic
go
lobe.
to our FlyComp website.
So we have a website like this. So to go to Newprint and Neuroglancer, all you do is you click on these links. It'll take you to those data sets. You'll need a Google account, by the way, to access the data sets in Newprint. So that's there to do. And yeah, so we've given a very comprehensive guide to how to, find your neuron and how to go about it. But very, very quick gist of this is let's go to this is my demo neuron, LC10A. And there are 140 specific LC10A neurons. But we'll just have a look at one. So we look at the purple one here. Right now I can see it's floating in this brain, but to make sense of this, let's extract a bit more features about it. So if you go to the side, we can click on this thing called ROI, and it gives us the layers of the visual system. And we call these layers neuropils because these are where most of the axons and dendrites of neurons are localized in these layers. And you can actually visualize these by stains, by microscopy data. So this is like an inherent biological structure of the visual system. If I press Control and I, zoom in, I can see like it's clearly in some layer here. So just FYI for you guys. So in school or you probably learned about how neurons look. You've probably, seen there's a very particular site where its dendrites are, where it gets information, a very particular site its axon and axon terminals are. But fly neurons are not like that. They're sort of all over the place. It's soma or cell body. It's sort of kicked off to the side over here. And the neurons are within the neuropils. These neurites, my apologies. So there are 4 layers to the fly visual system. So this is the lamina. The lamina in the data set is cut off a bit. This is due to technical reasons. This is the medulla, this is the lobula, and this is the lobule plate. It looks like a plate. And these colors don't change, so you can even look at the colors and tell, but that's essentially it. And this is all in the guide, by the way. So I can tell my neuron LC10A is in the lobula. That's one layer information I extracted. There's also something else you can do here, which is pretty cool. Since I've narrowed my neuron down to the lobula, I can click on this thing called lobula. Oh, sorry, that is not the right one. LO layers, not columns, layers. And if you click here, it gives me all the 7 layers of the lobula. And I can, from this, I can deduce to which layers these arbors are found in. So if you click on all of them, it will sort of cover everything. We have to peel one by one to see. So are there any arbors removed? No. Let's try 7. Are there any arbors removed? No, this is just the, I forgot what this was called, but it's just the tract that leaves the soma. So this doesn't have the arbors in it. These are not where the dendrites and axons are. And if we remove layer 2, we don't see any arbors showing, so it's not in layer 2, layer 6. You see, I exposed something right here. These are the arbors. These are like where the synapses and everything are going to be. I'll put layer 6 back on. Now let's remove layer 3. You see, even layer 3 has exposed some of these here. So we put layer 3. So from this exercise, I find out that my LC10A neuron goes through layer 3, 4, 5, 6. So I have 2 information now. It's in the lobula, and it goes from layers 3 to 6. Now, there's something else you can do in Neuroglancer, which you can see the sites of pre-synapses and post-synapses. So if I click on this toggle here, I can see it receives most of its inputs. Sorry, not pre-synapse means it's confusing. Pre-synapse means this is presynaptic to another neuron. It's sending outputs. So these are the regions, these are the synapses where it's sending outputs from. And you can click on post-synapse, which is here, which means where it's getting inputs. So it's getting inputs all over here. So that's something. One thing to keep in mind with neurons such as LC10A, this is a visual projection neuron, which is something you can see in the new CNS dataset. But unfortunately here you see this, it's truncated. This actually projects somewhere to the brain over here, but you can't see in this dataset. But if you go on to the male CNS, you'll be able to see it. So yeah, this is something, but for our competition, this is what we're using, because at the time this is what was available, but it's still a very helpful resource. And now let's have a go at Newprint. We can now search our neuron, LC10A, and we just click submit. And I can see exactly, these are the many IDs. Remember I said these are IDs, but this is a cell type. So they're being grouped by people. These are the IDs of the neurons. There are 140 of these. This tells me, which brain region these neurons are in. So it's getting its inputs from A02U. So remember we looked at A02U008. So it's actually getting its region from this brain region. And it's getting its input from a lobula. And it's sending its outputs to these brain regions. Neurotransmitter, it has is acetylcholine. And it shows you the number of inputs it's getting and the number of outputs it's sending. And if you click on this C thing here, you can actually see the sundial plot for this particular LC10 neuron. And essentially it tells you upstream who's giving this inputs by how much. So it seems like T2A, TMY21, TM4, TM5Y are all giving it its major inputs, and downstream we can see who's giving it outputs like LC14A1, MELO13, and so on. And if you notice, if you click on different ones, these will change. There's they connect to the same things, but the preference might be different. But if you actually analyze the data, this is something actually I do a lot, you'll find like overall most neurons will have a preference for one top one input, one top one output, and so on. So yeah, this is another level of information you can get from Newprint.
We have a question in the chat. Can you explore a sexually dimorphic neuron in the optic lobe?
So the optic lobe is from this data set, is from the male. You can look at the male version of it in the data set, but you won't be able to compare with the female. But you can have a look at this, and these folks have, organized it really nicely. So you can have a look at neurons over here, that dimorphic. You can specifically actually go to brain region visual system. So these might look familiar, medulla, lobula, and you can see like how they're dimorphic between female and male. You'll only be able to see, for example, TN26 will only be present in the optic lobe dataset. It's the male, but you won't find this in the female, and so on. So this is one way you can actually look at dimorphism. But the CNS is probably the best way to have a look at it.
I see. So for the visual system, you only have sex-specific neurons?
Do you mean you only have, or
Oh, like look, LOVP 105, that's dimorphic, which means they're different across males and females, right?
yes, they're the same neuron, but there's a slight difference in their
Great.
morphology.
Maybe if you can click into one of these, that would be nice.
Okay, sure.
TMY, maybe try TMY because I think we have
TMY looks very nice. So
So I guess
sometimes,
the point is that let's say you are exploring the optic lobe connectome and you see a neurons in the visual system and you're just curious how are they different across sexes. If that particular neuron that you chose is also sexually dimorphic or sex specific to the male, for instance, you can write that in your report as well.
yeah, yeah, absolutely. You can actually you go to this data set and say you've explored using this and you found this. That's totally acceptable. So you see, like the blue, I mean, visually, I personally cannot really tell the difference. This has been done through actually analyzing the data. So, you'll get like a table of neuron, the amount of synapses, blah, 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 and that's how people manipulate the data and extract this information. But visually, I can't really tell, but they're dimorphic between the male and female. So as you can see here.
Great. Thank you. I think it just looks cool to know that it's fascinating to know there's someone who took the time to actually explore the differences between these neurons across
Yeah.
data sets. So that's
Yeah.
very exciting.
If you're really curious, and there is a video on this by the authors of the paper, they've actually tracked I'm forgetting, the motor response circuit or something, all the way from the visual system all the way to the ventral nerve cord. So you can just follow how information travels. So that's really cool. You can do that too.
Great. We are approaching time, but before we go, Arushi, maybe you have mentioned a lot about the guides. If you could go to our website and show what those guides are,
Right.
that would
So
be
again,
great.
to the website, competition materials, it's up here. If you click on this. Anyways, so these are all our guides here. So we have our handbook. If you click on this, this is sort of the general sort of gist of the competition. And main details you can find here and deadlines. A starter guide we've given to give you essential information to get you started with the Fly visual system, how to explore neuroglancer. If you don't like watching YouTube videos, you can also follow the step-by-step guide over here. So that's there of Newprint and neuroglancer. We've given an example template. You don't really need to follow this, but it's a nice way to organize your research journey. And we've given a learning guide resource and compilation, about the optic lobe and like different sort of things that we didn't cover in the other one. So this is also very helpful for you to see.
That's excellent. And keep in mind that all of this we have gone step by step in workshop one, which you can access through our website. Yes, and as always, if you have any questions, please don't hesitate to ask through Discord or through our email, and all of that is also on our website. That's great. Is there anything else, Arushi?
Yeah, that was a lot of fun and really excited the male CNS is out. It's just if you like hear a lot of neuroscientists talk about this or like in the field, they at one point this was impossible. It's sort of a bit like science fiction that it's even here yet. So it's very, very exciting that we're able to even map brains to this level. So yeah, it's a lot of fun.
Excellent. Okay, I want to say a few last words, which is thank you, the audience, for showing us here, and thank you, Arushi, as well, for presenting the work. And yes, we will conclude the session today. Thank you.
Thank
Okay,
you.
bye.
Clematis Research Empowerment Hub. Empowering research for everyone.